Steps of PCR. There are three main steps of PCR which are called as denaturation. Second step is annealing, and the third step is extension, which is also called as elongation. So PCR consists of three main steps. First one, denaturation, in which the double-stranded DNA is converted into single-stranded. Second step is annealing, where the primers they bind to the single-stranded DNA, and the third step is extension, which is also called as elongation. In this step, DNA polymerase extends the newly strand that is complementary to the template strand. The first step in case of PCR is called as initialization step. So this step consists of heating the DNA which is double-stranded. When we heat the DNA between 93 degrees centigrade to 96 degrees centigrade, the double-stranded DNA is converted into single-stranded DNA. So usually during the first step, which is called as initialization step, at this step DNA or the tubes which are kept in the PCR machine, they are kept for approximately 1 to 10 minutes. So this is the first step in case of PCR reaction. So once the initial denaturation has been done, so first a regular cycling of the PCR starts, and the first step is again denaturation. During the first cycling step of the PCR, which is denaturation, at this step DNA is kept at 93 degrees centigrade, but there can be some variation. It can be 93, 94 or 95 degrees centigrade for 20 to 45 seconds. So during this step, the double-stranded DNA separates into single-stranded DNA. So this step is actually when we have to convert double-stranded DNA into single-stranded DNA. And that step is called as denaturation of the DNA. The second step of the PCR is called as annealing. At this step, which is usually at the temperature of 40 degrees centigrade to 65 degrees centigrade, there can be variation between the annealing of different primers. At some of the primers, they can bind at 40, 45, and usually maximum temperature of the annealing is kept up to 65, or in some cases it can be 67. So we can say that there is some variation in case of annealing temperatures. So annealing temperature, because during the previous step, the DNA which was double-stranded has been converted into single-stranded. During this step of annealing, the primers, which are also called as oligonucleotides, they will bind to the single-stranded DNA. This is the third step of the PCR, which is called as extension. In some literature or in some books, it is called as elongation. So elongation is the step where DNA polymerase adds the nucleotides and new strand is formed that is actually complementary strand of the template DNA. The polymerase has the optimum activity between 70 degrees centigrade to 80 degrees centigrade. But most of the time, or in most of the cases of PCR, commonly 72 degrees centigrade is kept, at which there is an optimum activity of the DNA polymerase. So this is the third step of the PCR, which is called as extension. Extension is done with the help of DNA polymerase, and mostly the temperature is kept 72 degrees centigrade. Extension. This is the last or the final extension during the PCR, because sometimes there are unfinished strands of the DNA. So a final extension is given that can be 1 to 7 minutes, so that if there is any unfinished products, that can be finished. So during extension, DNA polymerase that have added the DNTPs to the newly synthesized strand. So extension is the third step, and once extension is done, there is the final elongation or the final extension. At this extension, 70 to 74 degrees centigrade, it can be for 5 to 15 minutes, but in most of the cases it is performed at 72 degrees centigrade for 5 to 10 minutes. Final hold temperature, this is the last step of the PCR, that is for a short-term storage of the amplified product, because the PCR has been finished once there is a final elongation. So at the end, for a short-term storage of the amplified product, this step is added, which is called as final hold temperature.